we can share this with folks who were either attending the other session or who cannot be with us at this time today. And again, without further ado, I want to get started. Our first presenter today is Tyler Nguyen, who will be talking to us about real-time assessments of caspase-1 mediated inflammation after repetitive traumatic brain injury and the role in long-term pain. Take it away, Tyler.
Thanks, Rebecca. Can you go to the next slide? So today I'm going to give you a little bit of tidbit on the project, one of the projects I've been working on regarding a traumatic brain injury and chronic pain from a perspective of inflammation. So as you know, TBI has been a big problem in terms of trauma within the United States and worldwide. And when an individual have a traumatic brain injury, there are two phases of traumatic brain injury. There's an acute phase and then there's a chronic phase. So the acute phase composed of primary injury that encompass cerebral hemorrhages, skull fracture. If you have a more severe type of TBI, to synaptic injury and neuronal loss. And then when you get to the chronic phase where you get these inflammatory responses, and this conmes within 24 hours and lasted up to a week. In some condition that would last to a couple of weeks. And what I want to focus today on is the inflammatory aspect of traumatic brain injury. And in this case, I want you guys to pay attention to the innate immune response, particularly the inflammasome caspase-1 axis that responds after the primary injury. So TBI itself can induce a series of signaling cascade that including the release of these PAMs and DAMs, the changes in iron for both outside and inside of the cell, and all of these signals resulted in either the regulations of the protein that participate in these inflammasome caspase-1 complex, or involved in directly regulation of the activity of the pathway itself. And in this case, when inflammasome activate in this, for example, here I have the NLRP3-ASC inflammasome complex. The activation of this complex induce the recruitment of pro-caspase-1, which been cleaved to release the active caspase-1. Active caspase-1 then eventually will perform two functions. And one of the functions is shown here is cleave and activate IL-1 beta, or it can cleave and activate a compound called gasdermin D. Gasdermin D is a protein that will, once activated, it will form pores on the cell membrane. And enough of these pores form on the cell membrane, this induces lysis of the cell, which causing cell death through the route of pyroptosis. IL-1 beta is being released and recruit peripheral immune cell to come and response. The two important mechanisms that I want you to focus on here is how NLRP3 is being activated. And so it is in inactive form, NLRP3 is a monomer. Once there's an activator, it causes a conformational change that allowed the recruitment of the ASC complex. This allowed the recruitment of pro-caspase-1 that form these Ferris wheel-like structure. If you look at three-dimensionally, and this induces the cleavage of pro-caspase-1 to form active caspase-1. Active caspase-1, if you look at below, activated by once binding to this inflammasome complex. Caspase-1 itself is also a monomer. Once binding, it will dimerize. Somebody music is on.
Everyone please mute. Thank you.
Once The music still on. Can you guys still hear me?
Yes, we can still hear you.
Okay.
I have to figure out how to mute all the participants.
Okay.
Apologies.
All right. After dimerization, caspase-1 itself will cleave itself at the ideal linkage. I think Joe music is on because his pictures still come up when the music is turned on. So the ideal linkage, once it's cleaved, it will activate caspase-1, and this caspase-1 will does its job and cleave and activate I-1 beta and gasdermin D. As soon as this caspase-1 is activated, it will inactivate itself by cleaving the CDL linkage, thus release the P20 and P10 small and large subdomain. So when people classically do Western blotting to measure the amount of active caspase-1, they usually look for the P20 and P10 quantification. Can you go to the next slide? So our model here, it's we base our model on a repetitive brain injury paradigm, where this animal has been induced injury 3 times once a week over the period of 3 weeks. And these animals have been, so I prepare a specialized cranial window that allows us to imaging through the skull. So this wouldn't, so the skull of this animal has been thin. It's been thin to the point where it's transparent. So if you see here on the image, the thin side is, you can see very clearly the vasculature and the brain tissue underneath versus the not thin side where it's very opaque. And the injury model that we're doing is control cortical impact onto the skull. And the skull, once it's thin, it is being protected with a layer of cyanoacrylate glue that help maintain its transparency and also help preventing bone regrowth. So this window could last up to 6 to 7 months post preparation. Just to show that the injury model paradigm works, we measure intraocular pressure of these animals 5 minutes right after each injury, and we found there's an increase in intraocular pressure in both the left and the right eyes after the injury. In where you compare to the sham animal, there's no change in the intraocular pressure. We also look at the morphology of the brain tissue, and we found that there's no change in the tissue itself because we aim for this injury to be a mild type of traumatic brain injury. It's more like a concussion. So our model that we're using here is a caspase-1 reporter mouse, in which with our collaborator in Loyola University, we developed the reporter mouse that has a luciferase construct that is permutated, where the C terminal and the N terminal is being separated, and they test multiple different putative reporter, and what they found that the gasdermin D, the GSDMD permuted reporter, was the best reporter, where they put it in between the C terminal and the N terminal. So what happened is when caspase-1 is being activated, it will cleave the gasdermin D, in turn also cleave this luciferase construct. This allow the cleavage of luciferase, allow the binding of the C terminal to the N terminal, does activate luciferase. So when we inject this animal with D-luciferin, the activated luciferase will enzymatically cleave the luciferin, and this reaction release a photon. So we can capture the photon through an IVIS imaging system. So there's a direct correlation between increase in the activation of caspase-1 and increase in activation of luciferase, thus increase in the amount of photon, which is the light that we observe. Can you go to the next slide? So what we did here is we imaged these animals over a period of different time point after the injury. We also did that with the data that I don't show here, is that we imaged them after each injury as well. But these time points are after the last injury. So all of these animals have been suffered 3 injury. what we found is that there's an increase in these caspase-1 induced inflammatory response at 1 day after the injury in both male and female animal. And this increase lasted up to 1 week. And it's a multi-systemic increase. We found that's the increase in the brain, in the thoracic area, which is the chest area, the abdominal area, and also in the paws area. We're interested in the paw is because if you look at the graph on the right side, we perform von Frey testing to look at pain threshold response. I only showed the right paw, but we also did the left paw as well. The reason we did the right paw because the injury that we did was unilateral, which is on the left side of the brain. However, we saw there is a decrease in pain threshold, both the right and the left paw. A decrease in pain threshold in von Frey testing means there's an increase in pain sensitivity. So it takes less force for the animal to feel pain. What we did next was we tested a compound called MCC950. MCC950 is a direct inhibitor of the NLRP3 inflammasome. If you remember the first slide, the introduction slide that I showed you, inflammasome activate itself when there's a binding of upstream activator, and it recruit the ASC subunit as well as it recruit pro-caspase-1 in order to for the pro-caspase-1 to cleave itself into caspase-1. And the activation of inflammasome, particularly NLRP3, it's the requirement for this monomer to have a conformational chain is require a hydrolysis of ATP into ADP. So what MCC950 does is that it binds directly to the site where ATP is binding. So it prevents ATP binding, thus prevent the activation of the protein itself. And the interesting thing about this compound MCC950 is that it can cross the blood-brain barrier. So it had been tested multiple times in different other neurological and non-neurological diseases, but this is the first time that had been tested in pain, and particularly traumatic brain injury induced pain. So what we did was we did the same experiment where we treat these animals with MCC950 after the injury. And what we found that there is no significant change in caspase-1 induced inflammatory response after the injury, both at 1 and 3 days, in both male and female animal. We also perform behavioral of these animals after we treat them with MCC950 post-injuries. And what we found was that after the third it attenuates the pain sensitivity in the MCC950 treated group compared to a saline treated group. And the pain sensitivity stay low compared to the other two groups up to 1 month after the injury and after treatment as well. So what we conclude here, if you go to the next slide, is that traumatic brain injury, especially repetitive mild traumatic brain injury, resulted in multi-body increase in caspase-1 mediated inflammatory response through using the caspase-1 reporter mouse, luciferase reporter mouse model, and in vivo imaging. And what we also found was that pain threshold also decreases, thus more pain, and it lasts up to 2 months in both male and female animal. But when you block this pathway, particularly block NLRP3 activation, thus prevent caspase-1 activation, it attenuates this inflammatory signal, and it also help relieve some of the pain that the animal felt. So what the take-home message here is that this reporter mouse allow us to look at inflammation in vivo over time, which is something that has not been done very much in the past. Because when people look at inflammation in the past, you either have to extract the tissue from the animal, or you have to remove the blood and look at these for these inflammatory factor. So this reporter mouse allows us to look at in vivo in real time. And also what we found is that by interfering or modulating this inflammasome caspase pathway, we can modulate both the injury-induced inflammation and as well as its resulted pain response in the long term. Next slide. I'll take any question.
Tyler, thank you so much. We have time for one quick question if anyone has. Okay, I'm not seeing any or any in the chat. Tyler, thank you so much for a lovely presentation. I'm sorry about the
no
little bit of noise interference till I remembered how to mute. If there are no questions, then we'll proceed to our second presentation and thank Tyler again. So our second research blitz is from Nicole Weaver, and she's going to be talking about the role of GLDC in the embryonic kidney. Nicole, take it away.
Hi, everybody. I'm a part of Dr. Rebecca Wingert's lab. Super excited to talk about my research a little bit today. So you can go ahead to the next slide. So my research is looking at GLDC, which is a key enzyme in the glycine cleavage system, which you can see over on the left. The glycine cleavage system is made up of these 4 main enzymes designated by these kind of circular structures. Now, the P protein I have in the red asterisk is called glycine decarboxylase, or GLDC. Its main function is to convert glycine into these 1-carbon units to be later utilized in folate metabolism. Go ahead and click. So when GLDC is dysfunctional, it has been linked to neural tube defects and also the rare disease non-ketotic hyperglycemia. And this schematic gives a nice view of what's going on inside the animal when GLDC is dysfunctional. So on the left in the blue is GLDC in a wild type animal when it's functioning, versus on the right when it's dysfunctional. It's unable to break down those glycine units. We see this massive accumulation of glycine within the synapse and the synaptic cleft. The implications of this excess glycine are still being parsed out, but that's part of my research, is studying that accumulation of glycine within the animal. Go ahead and click. So when we first started studying this, we wanted to kind of see what the expression profile looked like of GLDC within the mammal. You can click again. Sorry, lots of animations. So one area we found expression was within the CNS, which is what we expected. However, interestingly, we also found expression within the kidney as well. So this is in the embryonic mouse, and we also saw expression within the kidney within the embryonic human as well. However, I didn't have space to show that data today. So go ahead and click to the next slide. And so we saw these expression patterns within the mammal. And so we wanted to kind of see what it looks like within the developing zebrafish, as that's the model that we utilize within our lab. So we looked at several different time points. Around 15 somite stage, we see this broad expression of GLDC, including the CNS and these kidney progenitor cells. At 28 somite stage, around 24 hours post-fertilization, we also see that heavy expression within the CNS. And then we also see expression pop up within the distal late segment of the embryonic kidney. So we see that expression continue throughout development. At 3 days post-fertilization, that same heavy expression within the distal portion of the kidney, and then also expression pop up within the neck of the kidney as well. And so we hypothesized, because of this conservation of expression and also the consistency of the expression within the kidney, that GLDC is vital for nephrogenesis. You can click on this slide, there's another portion. So in order to interrogate this a little bit further, we utilized morpholino oligonucleotides in order to disrupt splicing. And so my specific morpholino is targeting the junction of exon 1 and intron 1 and the junction of intron 1 and exon 2 that blocks splicing and leads to the retention of a premature stop codon. So you can go ahead and move on to the next slide. And so really quickly, I'll talk about why we utilize the zebrafish to study kidney development. So on the bottom, we can see schematic of a human kidney, which is made up of about 1 million of these functional units called nephrons, in which you can see all the different colors that are all the different segments that have various functions. On the top is a zebrafish at around 24 hours post-fertilization. We can see that it has 2 of these nephrons that run parallel down the length of the animal, which makes it a very simplistic model for us to actually study and look at the specific segments and changes that may occur. And so additionally, I'd like to emphasize that nephron segmentation is conserved across vertebrates. So hopefully with the color coding, you can appreciate that a lot of the segments within the human nephron are seen within the zebrafish nephron as well. So you can go ahead and click on this slide. So there's two specific segments I'm going to focus on today are this distal early and distal late segment. You can click again. So on the left is the distal early segment. On the right is the distal late segment. That top row of animals are wild type animals. In the middle are our GLDC morphins, so where GLDC is knocked down. And the bottom is GLDC knocked down, but then addition of scRNA. So first I'll focus on those middle row of animals. And so we can see that we do see a change in both of these segments within our GLDC morphins, and the quantifications are on the right. So within the distal early segment, we see that there's this increase or expansion of this segment. However, within the distal late segment, we see a reduction. And both of these phenotypes can be rescued with scRNA supplementation, which just verifies that our tools are specific. You can go ahead and click. So that's just the summary that we do see an expanded DE and a reduced DL. You can go ahead and click to the next slide. And so when we're thinking about development, a previous lab member, Dr. Chambers, she's made this really nice schematic thinking about how we start with these renal progenitor cells that then lead to specified precursors and eventually differentiated epithelium. That previous slide, I was looking at differentiated epithelium cells. And so what I want to do is I want to go back up that pathway and see where GLDC fits into the pathway of nephrogenesis. You can go ahead and click. So to kind of start teasing away that pathway began by looking at this transcription factor, IRX3B, that then leads to cells of the distal early fate. And so what we saw was when we knocked GLDC down, we see this expansion of the IRX3B domain, which if you recall on the previous slide, we saw that the distal early domain expanded. So this makes sense, right? We have more cells expressing IRX3B that then become those distal early populations. Go ahead and click to the next. So just wanted to give a quick summary of what we talked about today. So we found that GLDC is expressed within the developing zebrafish kidney, similar to what has been seen both in mice and humans. We also found that upon knockdown, GLDC morphins exhibit segment changes. And specifically, I was focused on these distal segments today. And lastly, that GLDC morphins exhibit changes in transcription factors that are responsible for renal cell specification. So for future directions, a lot of data I wasn't able to show today, but specifically, I'd like to investigate more about the cellular dynamics of what's going on within these distal segment populations upon GLDC knockdown. And then additionally, like I stated in the first slide, GLDC deficiency has been linked to neural tube defects and non-ketotic hyperglycemia. So I'd really like to be able to treat my animals with bioactive or FDA-approved drugs to see if we can reduce or eliminate any of these developmental defects. You can click to the last slide. And so I just wanted to say thank you to the funding, my advisor and lab, and I'll take any questions if anybody has any.
Okay, thank you, Nicole. Do we have any questions from the audience? Okay, I'm not seeing any questions. Nicole, thank you so much for a nice, clear presentation. Thank you. So now we will go to our third presenter. And this presenter is Kaylee Cloghessy. She is a graduate student in the Hyde Lab here at Notre Dame. And Kaylee will be talking about the genetic mechanisms that underlie regeneration within the zebrafish, specifically in the Telencephalon following traumatic brain injury, and her approaches using RNA-Seq. So Kaylee, I'll give the floor over to you.
Thank you so much, Rebecca. As Rebecca mentioned, my name is Kaylee Cloghessy, and I'm a PhD candidate here at the University of Notre Dame. And our lab, the Hyde Lab, focuses a lot on the genetic mechanisms that underlie regeneration after injury in the central nervous system. So today I'm going to tell you a little bit about the approaches I'm using to study those genetic mechanisms after traumatic brain injury, or TBI. So Rebecca, if you'd please click. But first, I want to give you a little bit of background as to the importance of studying TBI, and that's really that anyone's at risk for getting a traumatic brain injury at any point in their life. And in the US alone, there's about 1.6 million TBIs that occur each year. So it's a relatively large problem. And today I'm going to focus mostly on blunt force TBI, which is a strike to the head, or your head hits an object. So if you think a car crash, and this is the most common type of traumatic brain injury that we see in the human population. Rebecca, if you'll please click. And a traumatic brain injury can lead to a whole host of different long-term side effects, like cognitive issues, behavioral issues, and even physical manifestations. And given the fact that we have these detrimental side effects and they're so common in the human population, there's actually a surprising lack of treatment. And Tyler gave us a really good background on both the primary and the secondary injury. So I'll just reiterate that the primary injury occurs kind of as a result of the mechanical force, and it can lead to cell death and a bunch of other, biological processes. And the biological processes that become chronic can contribute to what we call a secondary injury, which can lead to lasting cell death. And it's often, treatment actually is often focused on managing that chronic cell death versus, treating and trying to regrow maybe the cells that we have lost. And this is because mammals, like humans, don't really have a huge capacity for adult neurogenesis. So it's often thought that those neurons are kind of awash, they're lost after injury. So we kind of thought, okay, could we use a model organism that maybe has a robust regenerative capacity to determine if we could find target genetic pathways that could potentially be modulated in humans after traumatic brain injury to kind of to jumpstart endogenous regeneration. Rebecca, if you'll please click. So we proposed the zebrafish, and the zebrafish, like I said, has a robust regenerative capacity, but it's also actually got a relatively similar genetic and morphologic homology to other model organisms like the mouse. But we can see this robust regenerative capacity in the first row. These are sections of the zebrafish Telencephalon, and the red is PCNA, which is a marker of proliferative cells. And you can see that even in the undamaged, they have some basal level of proliferation. However, if we stain for PCNA in fish that have gotten a severe traumatic brain injury, at 72 hours, we see a significant increase in the amount of those PCNA positive cells. And I didn't have time or space to show the data here, but we've actually shown that some of those progenitor cells actually become mature neurons a little bit after injury. So Rebecca, if you'll please click. So what are these underlying genetic mechanisms and how do we plan to assess them? So we're going to take an unbiased approach here using a tool we call single cell RNA-Seq. And basically what we're going to do is we're going to use the model for blunt force traumatic brain injury, which is a modified Marmoro weight drop where we drop a weight on the zebrafish head, and that delivers the blunt force traumatic brain injury. And our focal point is right at the telencephalon. Now we chose the telencephalon mainly because it contains the analogous region to the human hippocampus, which is the center for learning and memory. So we're going to dissect out those telencephalons at various time points after injury that correspond to different aspects of that regenerative process. Then we're going to dissociate them into single cell suspensions, and we can prep them, sequence them, and then analyze what genes are turned on and which pathways are turned on at different time points that correspond to these different regenerative processes. Rebecca, if you'll. So we have some preliminary results here. This is a U map shown on the left with all the colors. For people who are unfamiliar with looking at UMAP or single cell analyses, basically what happens is each small tiny dot on that graph corresponds with a cell, and we can group cells based on how similar their transcription profiles are. So cells that are very close together are very similar, and cells that are further apart are transcriptionally different. And basically we can cluster those cells, and then we can give them, based on their gene expression, we can assign them to the different cell types of the CNS. But further than that, we can actually go in and we can look for expression of our proliferative marker, PCNA. So if you'll see there kind of at that little lip, you can see an increase, yep, exactly, in that PCNA expression, which corresponds to progenitor cells that are very closely related to radioglia. So furthermore, we can actually go in and in that specific cluster, look for other genes that are upregulated, which are going to be our target genes. So for example, pulling out FGFR2, which has actually already been shown in a couple of other model organisms to be related to neurogenesis. So we can come up with these list of target genes, and then we can modulate them to determine their exact role in vivo in the zebrafish in regeneration. So then Rebecca, if you'll, yeah, so our conclusions are we can perform the single cell RNA-seq and generate these unique clusters of cells, which we can give cellular identities. We can then determine which cells are proliferating and which other target genes are expressed in those proliferating cells. And then we have just started actually our big project in this, which is we're going to compare the differentially expressed genes across different actually damage severities and different time points across regeneration. And through that, we can kind of tease out these different genetic mechanisms. Yep, next slide. And then I'd like to give a quick shout out to my lab for being so supportive, of course, funding sources, and the wonderful people who take care of our zebrafish here at Notre Dame. And with that, I'll take any questions.
Kaylee, thank you so much. Are there any questions for Kaylee? Open the floor.
I do have one question regarding the zebrafish TBI model.
Mm-hmm.
How do you establish that the TBI has been induced? You know, because in rodent, you can do various measurement, intraocular pressure, behavioral measurement to look at the sequelae of the injury. In the zebrafish, how do you know that the injury actually has some sort of similar effect?
So for individual fish, like fish
Yeah.
I would then use to
Right.
go on to the single cell RNA-seq, we do see, especially because this is a severe injury, we see a lot of subdural hematoma. And so that's one way that we can measure that. However, we actually just recently published a paper on this model. We've actually gone through pretty extensively and tested breakdown of the blood-brain barrier. We've tested behavioral aspects. We've tested cell death with like a tunnel stain. So normally if I'm using a fish for something and I can't kill it, obviously if it's going on to be used in a single cell RNA-seq, I would look at disruption of the blood-brain barrier, or if that fish winds up having seizures, if the fish in the group have a death rate that's consistent with what we see in the severe TBI. So more stuff like that.
Okay.
But if it's just testing the model, we have.
Okay. So the animal does survive the injury.
Yes.
Okay.
At about 50% in a severe
okay.
group.
And it's right. So like I said, it's equivalent to a severe TBI in a high order animal. Okay. Okay. Because I very rarely see TBI. I think in all of the conferences I've been to, your talk was the third talk that actually used TBI on a fish that I've seen. So interesting. Thank you.
Right. If there are no further questions, then we'll thank Kaylee again for a lovely presentation. Thank you, Kaylee. And we will move to our last presenter of this Research Blitz session. And so here we are going to hear from Gowthami Mahendran, who will be talking to us about unraveling the pathways in Miller-Dieker syndrome through gene expression analysis. And Gowthami, I'll give you the floor.
Thank you. Hi everyone, I'm Gowthami Mahendran, and I'm a third-year grad student in Dr. Jessica Brown's lab. And today I will be talking about my project titled Unraveling the Pathways Affected in MDS through the Gene Expression Analysis. So Miller-Dieker syndrome, in short, is MDS. It's a brain malformation. So the figure down there, it shows the human chromosome 17, which is normally associated with genomic instability that can be linked to many rare neurodevelopmental diseases. So the chromosome 17p13.3 region, it includes the MDS locus, which spans around 26 genes, which are shown in there. So the outermost genes, the YWHAE, it increases the severity of this disease, and also the deletion of that causes increased severity. And the other one, the PAFAH1B1, the other corner, yeah, it's responsible for the characteristic signs of lissencephaly upon deletion. So, so the severity of this disease is based on whether the whole locus is deleted, including the major genes, or whether some of the genes are deleted. If the entire locus, including those two genes, are deleted, it causes the infants to die in utero at 10 to 20 weeks of post-gestation. And if they don't have this much of deletion and only some of the genes are deleted, then they do survive with the following characteristics, which are listed below. So agiria, which means unusually smooth brain. So the figure on the right top corners, it shows the normal and the lissencephalic brain. So the normal brain, it normally has a lot of folds in it. But when you compare it with the lissencephalic brain, it has a much lesser fold in the outer appearance. So which means unusual smooth brain in this type of a disease. And another characteristic is the microcephalic, which means a small head when comparing to the normal individual. So you can compare you can see the children who are affected with MDS. And other than that, they do exhibit some other characteristics like intellectual disabilities. They show growth retardation and impaired motor abilities. And one thing I forgot to mention is this deletion is a heterozygous deletion, and it occurs in the chromosome 17 2.8 megabase pair region. Yeah, next slide. So this is the workflow. So in my in vitro study, I leveraged two different cell types, the BJ cells for the normal control, and the GM06097 cells derived from the MDS patient skins, so as my disease control. So the fibroblasts extracted from these both cells, we did an RNA extraction and a protein extraction. So followed by the RNA extraction, we did the RNA sequencing, and we did the differential expression sequence analysis and several pathway analysis using the IPA, gene ontology, and the Panther classification systems, which are shown on the top. Yeah, that one. And further, we are also going to do some alternative splicing analysis using some softwares, which I have not mentioned in this. And now currently we are in the step of validating it with the RT-qPCR for the selected genes from RNA sequencing. And in our proteomics study from the protein extracted, we did a tandem mass spectrometry analysis and did several proteome analysis. And then we validated these results with the Western blot studies. Yeah, next slide. So from our transcriptomic studies from the RNA sequencing, so within the Miller-Dieker syndrome locus, the ones highlighted in blue are the downregulated ones. So we found most of the genes to be downregulated within the MDS locus at the RNA level, and only one was found to be upregulated. And the volcano plot down there is showing the up and the down regulated genes. So in total, 1,286 genes were found to be upregulated and 1,115 to be downregulated. However, this trend was not similar to what we found from the proteomic study, which is shown on the right. Yeah, so we found only 4 of the genes to be downregulated and 2 of the genes to be upregulated, colored in red. And the one in black are the ones which have not shown any significant alteration. So at this point, we did several, can you go ahead and play? We did some pathway analysis. So this is the GoTerm analysis that we did just to look at which pathways are having some interesting genes which are getting altered. So we selected the ones with that are shown in boxes, the skeletal system development, synapse organization, which are involved in neuronal system development, then some of the organ development, renal system, kidney development, sensory organ morphogenesis. So from some of these pathways, we selected the most interesting genes, and then we validated it with Western blot, which is shown on the right. So these are some of the genes that we tested, which are associated with the smooth muscle contraction, protein ion transport, actin binding, antigen processing, and several cellular localization processes. Next slide, please. So from our studies, we are concluding that we are seeing a significant alteration in the genes at the transcriptional and the translational level, including the pathways which involves the synaptogenesis, skeletal system development, organ developments, motor regulation, and RNA binding. And also our proteomics data validation results from Western blot studies, it indicates that there's a compromisation in muscle contraction and transport of ions and proteins in the MDS patients when comparing to the normal control. And now currently we are in the state of validating the genes which we found to be significantly altered in the transcriptional translational level using the qRT-PCR and the Western blot. And our future directions will be to look at the splicing related proteins which can get altered in the MDS from our proteomics related studies, because splicing is one of the important things that gets altered in this kind of a neuronal disease. Yeah, with that, I thank everyone who are here listening to my talk, and I'm happy to accept any questions.
Gowthami, thank you so much for a very lovely talk. Let's open the floor, see if there are questions from the audience. Okay, well, wait to see if anyone, oh, I see there's some clapping from the audience, some silent clapping with the icons. We're waiting to see if anyone else has questions. Of course, my curiosity is piqued by your changes that it looks like you're seeing potentially in kidney development. I don't know if you're prepared to talk about any of that or if that's for a later time. Either way is fine, but yeah, curious.
So we have not done any further studies, so we are just currently in validation step and looking at. So we have not done any with the kidney development thing. So yeah, soon I will be able to talk maybe next time.
Okay, yeah, so stay tuned. Thank you.
Thank you.
Okay, any other questions? Okay, if not, again, Gowthami, thank you for a lovely talk. Thank you to all the presenters for taking the time to deliver these great research blitzes. And I think we all look forward to seeing where your research goes. Thank you so much, and have a great rest of the time at the CTSI events today.
Thank you.
Thank you, everyone.